Androecium. As I told you, andro it means a male, andro means male, and ecium means house. So this is the male sex organ, and its units are called as stamen. And when we see the structure of stamen, the stamen consists of two parts, that is anther and a filament. Here you can see, here is the this one part, this part is anther, and the long slender part is filament. This one is the filament, this one is the filament, this is sterile part, and this one is the fertile part. Then when we take the transverse section of anther, we can see this is a dithecous anther. Di means two and theca it means a chamber. So when we take the section, so we can see it's after taking its transverse section, we can see these four locules chamber. Like if we see the vehicle in a horizontal way, we can see only the two tires, but actually it has the two tires on the either side. So like that, and if the dithecous anther after taking a transverse section, we can see the four locules, and these are the four locules, that is tetralocular. And after taking the transverse section, we can see this part after taking transverse section. So the anther will show this part, that is from the outer part is epidermis, which is protective. Generally these three layers are protective, that is epidermis, endothecium and middle layer. These all three layers are protective in function. So this epidermis is outer most protective in function. This endothecium, it is hygroscopic. Hygroscopic means moisture absorbing. It has the moisture and have alpha cellulosic wall as well. Then these are if you see this epidermis, endothecium, here you can see single layers are there. And in the middle layer, that is 1 to 3 layer structure, it is epimeral. Epimeral, the word is used as here for middle layers is epimeral. Epimeral means it is short-lived, not staying for the long time. It is short-lived because during dehiscence it will burst and pollen grains are going to release. Then the innermost layer is called as tapetum. Tapetum is the innermost layer, which is nutritive. Its cell contain more than one nucleus, that is they are the polyploid in nature. So tapetum, and here is the connective, means here we can see the connective, means which will connect this filament is going to connect to this anther by means of connective. Here is the connective tissue is there. So this is anther. Then the here this is the diagram of transverse section of mature anther. Then enlarged view of one microsporangium. Name is given here microsporangium. Micro it means small, and spora it means spore, angium it means bag. This is called as microsporangium also, because it contains the microspore. So that's why the name is given microsporangium. You see the structure of this tapetum. In the tapetum, there are the two kind of cells are there. One is called as secretory cell, and another is plasmodial or invasive cell. Secretory means, as the name suggests, it will produce a secretion. Which kind of secretion? It will produce a secretion which will help for the pollen germination. So, which kind of secretion it will produce? It will produce, it has pollen kit, then sporopollenin, and then compatibility factor. Compatibility factor. These things are going to use for when pollen grain land on the stigma. So during the germination of this pollen tube on the stigma, these factors are going to helpful. Not all, but if you see this sporopollenin is the most resistant biochemical which is present on the enzyme of pollen grain. Pollen kit is present, then compatibility factor is present, and plasmodium or invasive cells. So being the tapetum, it is having the thick and dense. It has thick protoplasm which dissolve and provide the nourishment to the developing that microspore. And in the anther there are the microspore mother cell. So after meiosis, that is reductional division, they will produce microspore. Microspore mother cell undergo meiosis or reductional division, and they will produce the microspore or pollen grain. Then, so if we see the cell division in the pollen grain, then there are the two kind of division. That is, one is simultaneous, another is successive division. In the this kind of division means the successive and simultaneous. The difference is that in the simultaneous, the division is going to take place, this division of cytoplasm, that is cytokinesis, is once, this at the end, and here the cytokinesis is going to take place at two different places. If we see the pollen grain, it is going to produce in the anther from the microspore mother cell after the meiosis. After meiosis, this pollen grain going to produce. In the anther, there is the microspore mother cell. Where it is present? It is present in the anther. So after the meiosis or reductional division, this microspore mother cell is diploid in nature, having two set of chromosomes. So after meiosis, it will produce the haploid microspore. It will produce haploid microspore or pollen grain. And from the one megaspore mother cell, the 4 microspores are going to produce. That is, tetrad is going to produce. And when we see the structure of this pollen grain, so this is the pollen grain. When we see the structure of pollen grain, it has its outer wall called as exine. Exine, EX, external, outer. So exine is outer layer of this pollen grain, and it is made up of a chemical called as sporopollenin. And in the biological world, the sporopollenin is the most resistant chemical because it cannot be affected by the action of strong acid, alkali, or none of the enzyme discovered so far which can harm this exine. And because of this reason, the pollen grain can be found in the fossil form. Study of pollen grain is called as palynology. But because of sporopollenin in the exine, it can be found in the fossil form also. Then this number 2, this is intine. Intine is the inner layer of pollen grain, which is made up of pectin, cellulose is there, and exine. See, exine is outer layer and not continuous. It will be showing germ pore between here. Germ pores from where the pollen tube is going to come out. Generally, this germ pore, if it is present only one germ pore in a one pollen grain, so that is called as monocolpate. If there is one germ pore in a pollen grain, this is the pollen grain, and if only one germ pore is there, then that is called as monocolpate. If there are the two germ pore, it is called as dicolpate, bicolpate. And if there are the 3 germ pores are there, it is called as recolpate, RICU, recolpate. Generally, monocot have one germ pore and dicot have more than 2 germ pores sometimes, or 2. Then this intine is smooth, exine is not smooth continuously. Then, so after the intine, here will be a plasma membrane. Then inside it we can see the cytoplasm. Then there is the nucleus. In the microspore, the smallest microspore is from myosotosis, whose size is around 2.5 to 3.5 micrometer. And if you see the largest pollen grain, that is microspore, is from mirabilis, that is Mirabilis jalapa. So size of that pollen grain is around 250 micrometer, and the longest pollen grain is from the Zostera. Zostera is the only marine angiosperm, whose size is around 2500 micrometer. Average size of micrometer is average size of microspore is between 25 to 50 micrometer. And already in the structure of microspore we saw it consists of outer layer, that is exine, made up of sporopollenin. Intine is made up of pectocellose. Then cytoplasm is there, and centrally placed nucleus, and plasma membrane is also there, and the germ pore. The structure already I discussed. Then the formation of microspore from the microspore mother cell is by meiosis. So that is called as microsporogenesis, formation of microspore from microspore mother cell. And from the microspore, when the two male gamete is going to form by 2 mitotic division, so that whole development is called as development of male gametophyte. Average size of pollen grain is around 25 to 50 micrometer. Pollen grain is a microspore which is produced from the microspore mother cell that I told you. So when the development of this microspore is going to happen, that is when we see about the development of male gametophyte. Development of male gametophyte. Here we can see the development of male gametophyte. So as maturity is going to arise in the flower, so what will happen? The pollen grain is going to develop. So after the mitosis from the pollen grain, when we saw the structure of pollen grain, see here the pollen grain is having different kind of sculpture on its outer layer, means this one outer part, that is egg sign is there. Then pollen grain after the mitosis, it will produces the two cells, that is one is vegetative cell and another is generative cell. So here we can see, here is the germ pore is present. The information is already I given about germ pore. This is pollen grain which has germ pore, then egg sign in time. So after mitosis, it will produce the two cells, because in the mitosis we know it is equational division. Vacuolar nucleus is there. Then it will produce the two cells, that is one is vegetative cell which is large. This cell is large and this one is small cell. Vegetative cell which is irregularly shaped, and vegetative, I told you the word veg, it means here food, and being it is going to store the food, its size is big. Then generative cell is small. So as the name suggests, generative cell, so it will generate two male gamete after mitosis. So that's why the name is given generative cell. So generative cell again undergo the mitosis to produce the two male gamete. Here we saw this vegetative cell and generative cell is present. Then so after mitosis, this generative cell will produce the two male gametes. Two male gamete. Here two male gametes are shown. This is one, this is another. So two male gametes are produced from the generative cell by mitosis. So where the mitosis take place? See here, at the two places, from the pollen grain, after the first mitosis, that is after the first equational division, one vegetative cell and generative cell is formed. One vegetative and one generative cell. And after another mitosis, means number 2, after mitosis, second mitosis, from the generative cell, two male gametes are formed. Two male gametes are formed. Generally in the 60% of the plant, the pollen grain is released at the two cell stage. Two cell stage means what? This stage is two cell. Which cell is C? This is two cell stage. This is two cell stage of pollen grain. That is vegetative cell and generative cell. So in the angiospermic plant, 60% of the plant, 60% of the angiospermic plant, the pollen grain is shed, means it is released from the anther at two cell stage. And in 40% of the plant, the pollen grain is going to release at the three cell stage. Three cell stage means one generative cell and two male gametes. Two male gametes. So this is three cell structure. Three cell means what? One vegetative and two generative cell. So at this stage, the pollen grain is shed from the anther. And already I shown you the structure of that anther. So it will show that lining of dehiscence. Like you can see in the capsicum, there are the lines of dehiscence from where it is going to break down. Because its layer that I told you, that endothecium is hygroscopic. It will absorb the moisture, because of which they will show the rigidity and that maturity. Being the water is going to lose, so it will dehisce, means it is going to broken down, and the microspores are going to released, and then they are transferred by means of pollination. Then this is about the pollen grain. Then if we see about the pollen viability, viability means how long they can be in a good condition. So that is called as viability. Means their viable period, still they are going to stay alive. They are not died. They are in working condition. So if we see in a plant like wheat or rice their viability is up to 30 minutes. 30 minutes. That is half hour. Half hour. And in some plant, like the family of some plant, like there is a leguminous is there, rosaceae is there, then solanacea is there, leguminaceae, rosaceae, solanacea. These are the family of plant. In this plant, the pollen grain can remain alive up to the several months. Several months. It can be present in that condition for several months in these plants. Then pollen grain, it is some people can have also the allergy for pollen grain because like here you can see Parthenium plant is there, carrot grass, or there is another plant is there, Amaranthus, the in family of Chinupodiaceae. This the flower of this plant can the flower the pollen grain from this flower can cause hay fever, HAY, hay fever. Hay fever. By Amaranthus, the pollen grain of this plant, Amaranthus, then Chinopodiaceae, so on. The pollen grain of some flower can cause allergy, like bronchitis they can cause, asthma they can cause, then hay fever can cause. These are the things which can be caused by the pollen grain of the plant. Which diseases? Like I told you, this hay fever, bronchitis, that is inflammation of bronchioles. Bronchi means bronchi in the lungs, and ITS word stand for the inflammation, that is red hot swelling pain, that is called as inflammation. Then it can also cause the asthma, that is shortness of breathing, that can be caused. And this is this plant came in the India as an contaminant with the wheat. It was the tactics of Britishers then. But there are some uses of pollen grain as well because they are nutritive. So it can improve the performance of athlete. The pollen grain come in the form of pollen tablets also. So that can improve the performance of athletes, horse, in case of horse. Then up to here, this part we saw the what is by microspore, pollen grain, and which kind of pollen grain are present. Then development of male gametophyte, then pollen viability, then what about the nutritive value of this from the nutritional aspect of pollen grain. Then we also saw which kind of disease can also be caused by the pollen grains. Then